Thank you for the invitation to present our work. So we just start at the beginning in terms of follicles being formed within the human ovary. And of course, that takes place before birth. The oocytes are formed, they enter meiosis, they're arrested at the dictate stage of prophase 1, and they form the group of primordial follicles that will be utilised throughout the female's reproductive lifespan. And of course, we know that the primordial follicles are initiated to grow at various times during our lifespan and leading to the formation of a mature follicle and mature oocyte that will lead to ovulation. Now, this has been an area of debate for quite some time as to whether the mammalian ovary has any capacity for regeneration, meaning can new eggs be formed after birth or in adulthood? And it has been an area for well over 100 years that scientists have debated, and there's been various evidence on both sides put forward. The debate was reignited in the early 2000s when the group of Jonathan Tilley at Harvard in the United States isolated cells from the mouse ovary that appeared to be oogonial stem cells, so they could produce new oocytes. And then in 2010, he continued in this work and isolated a population of cells from the adult human ovary where these cells appeared to be oogonial stem cells, so could form new oocytes. And the basis of these cells, or the characteristics of these cells, were that they expressed DDX4, which is a germline marker, and they could proliferate, which of course oocytes don't proliferate. From these cells, they could dissociate the adult human ovarian material, and then using a DDX4 antibody, they would separate out cells that had DDX4 on the surface, and the oocyte has DDX4 within the cytoplasm, but these cells could be isolated through DDX4 surface expression. And then they could take these cells and transfect them with green fluorescent protein, inject them into human ovarian cortex, and then transplant them to an immunological deficient mouse, to a xenograft. And when they isolated the tissue after between 7 and 14 days, they could see the formation of what appeared to be new oocytes, and they expressed GFP, surrounded by somatic cells that did not express GFP. So indeed, it looked like new follicles were being formed. Now, this led to a great deal of controversy because the isolation methods were based on using an antibody to DDX4. And this was controversial in that many people believed that DDX4 was not a surface, it couldn't be expressed on the surface. Well, that's been shown not to be the case. It can be expressed on the surface. But the initial experiments only used cells that had been cultured. So they, when they injected them into the tissue, there was an argument that it could have been an in vitro transformation. The physiological role or capacity of these cells have not been established. It's a rare population, so reproducibility in isolation has been reported in that many groups have been unable to isolate the cells and so have come to the conclusion that the cells don't exist. But several groups and papers have been published, or several groups have published papers, and they've isolated these putative oogonial stem cells from a range of mammalian species, including the human. And of course, if we look into the context of adult stem cells, this is a fast developing area. If we had been speaking, maybe even 10 years ago, we wouldn't have known about any of these adult stem cells in all of the organ systems of the body. So it's likely that the ovary does have stem cells for all ovarian cell components, but it doesn't necessarily follow that they're actively involved in forming new follicles or oocytes during adult life under normal physiological conditions. But of course, being able to isolate such cells allows us to look at the whole process of germ cell development in vitro in particular. So we've been interested in these cells for a number of years now, and we've developed a methodology to dissociate adult human ovarian cortex using FACS and using DDX4 as Tilly did. But in addition to DDX4, we use aldehyde dehydrogenase activity, which is an indicator of stem cell activity. It's an indicator of viable stem cells. So you'll only see it within intact cells, and if there's a high level of it, that's associated with the stemminess of the cell. And this has been published in scientific reports. So when we do this kind of sorting, we can define 3 separate populations of cells based on their size and their degree of aldehyde dehydrogenase activity. And when we look at markers in each of these populations, we can see there's germline markers and stem cell markers, but not follicle or oocyte markers. So therefore, we're not pulling down oocytes, as has been suggested previously. But when we look at each of the populations, we can see that they differ in that, so population 2, for example, has a splice variant of the DDX4, and population 2 also expresses DAZO, which the other populations don't. So the question is whether there are subpopulations of represent developmental stages of these cells. But these cells, what we've been able to do is we've been able to analyze them immediately. So take them before culture. So the markers that we are observing, so they're observing DX4, for example, both vitro in particular. So we've been interested in these cells for a number of years now, and we've developed a methodology to dissociate adult human ovarian cortex using FACS and using DDX4 as Tilly did. But in addition to DDX4, we use aldehyde dehydrogenase activity, which is an indicator of stem cell activity. It's an indicator of viable stem cells. So you'll only see it within intact cells, and if there's a high level of it, that's associated with the stemminess of the cell. And this has been published in scientific reports. So when we after we had isolated these cells, we thought we could combine them with somatic cell support to determine whether they could make oocyte-like structures. And we combined them with human fetal somatic cells. And our reasoning for that was because of the work that had been done on using induced pluripotent stem cells in mouse. And when they combined them with fetal somatic cells, they were able to form, more Oogonia-like structures. So that was the rationale. When we combined them with the human fetal cells, we had the formation of aggregates. And within these aggregates, we could see the formation of what appeared to be oocyte and follicle-like structures. And these follicle-like structures would develop into multilaminar structures with what appeared to be an oocyte structure within the middle of it. So it did look as if by combining the putative oogonial stem cells with fetal somatic cells, we could get new follicles being formed. And that's been published in scientific reports. So distinct populations of cells with germ and stem cell markers can be isolated from ovaries of adult women. And combining these cells with fetal somatic cells within an in vitro growth system leads to follicle formation. But we need to determine the optimal support. somatic cell support for oocyte development, because fetal cells is not really a practical solution. And also we need to learn whether adult ovaries have somatic cell support for the germline stem cell. Now, we would suggest that indeed the adult human ovary does have somatic cells that will support these booster oogonal stem cells. And if you inject GFP-labeled booster oogonal stem cells into human ovarian cortex, you can see that these cells develop into what appears to be oocytes and follicles. But they're clearly recruiting cells from within the ovarian stroma. So there are cells within the adult human ovary that will support the development of these germline stem cells, or the boost of germline stem cells. Now, the adult human ovary, if you've heard from previous speaker, Francesca Duncan, be speaking about structures within the ovary, it contains many different cell types and different structures. There's a component within the ovary that we still know very little about, and that's the stromal component, which is composed of several different cell types that hasn't really been well characterized. So in terms of the support or search for support cells, we've looked at mesenchymal stromal cells. So these MSCs, which are an efficient cell type that's been used in clinical applications of cell therapy, derived from several tissue sources, pancreas and bone marrow and other sources. There's several reports that indicate that these MSCs could promote the recovery of ovarian function, and ovarian MSCs have not been isolated yet. So you can see here that MSCs can be multi-progenitor and form different cell types. So the goal for us then, or our aims, was to determine whether these MSCs could be derived from adult human ovary, and whether co-culture with them could enhance the formation of follicle-like structures from OSCs, so providing somatic cell support. And also, we're interested to determine whether co-culture with these MSCs will have an effect on primordial follicle survival and activation in vitro, so within our in vitro growth system. So I'm just going to go through how we've established cell lines of ovarian mesenchymal stromal cells. Of course, our model is we use biopsies that are obtained from women, normally at the time of caesarean section. We also have other sources of tissue, but we can then take this tissue and dissociate it. And then, of course, we go through a whole series of filter stages, and then we culture them on plastic dishes in just regular media. So these, one of the characteristics of MSCs is that they're plastic adherent. So we're looking for the plastic adherent cells. The markers of the MSCs, as defined by the International Society of Cellular Therapy, is that they're plastic adherent under standard culture conditions. They have a surface expression of these factors, the CD105, 73, and 90. They don't express CD45, 34, 14, or CD11B, 79, 19, or HLA class 2. And that they can have, that they are multi-lineage differentiation potential in that they can produce osteoblasts, adipocytes, and chondroblasts. So that's the kind of minimal criteria to define whether the cells are indeed mesenchymal stromal cells. So we've carried out these testings on several, so we've taken our associated cells, put them onto plastic, and then taken them, passaged them, and then tested them for the markers and compared them to our pancreatic mesenchymal stem cells. So the pancreatics are red and the ovarians are blue or light blue and pink for the pancreatic cells. And essentially, for all the markers, they're showing a similar profile. The ovarian-derived cells are showing a similar profile to the pancreatic-derived cells. And then we've gone on to test the multilineage capacity of these cells. So essentially taking our cells and growing them in media that would support either a differentiation to osteoblasts, adipocytes, or chondroblasts. And that's, we would culture them and then test them by staining for each of these cell types. And when we compare them again with our pancreatic mesenchymal stromal cells, what we find is that essentially the ovarian-derived cells behave in a similar way in that they are osteogenic, chondrogenic, and adipogenic. So there's positive staining for each of these cell types in our samples from the ovarian-derived cells. So the summary then, cells with germline and stem cell markers can be isolated from the adult human ovaries, or putative OSCs. Ovarian-derived mesenchymal stromal cell lines that meet ISCT requirements can be established from adult human ovaries. So we have 5 cell lines derived so far. And then the next steps are, of course, a series of co-culture experiments to determine whether these ovarian MSCs can support the formation of oocyte-like structures from our oligonial stem cell cells, or putative OSCs. So the next step that we've done, and this was before we had fully characterized cell lines, we took our somatic cells. So essentially, when we fact sorted our OSCs, we took all of our negative cells and we cultured them just for a short period of time before combining them with the OSCs. So it would be like a mixture of cells that would, of course, include these mesenchymal stromal cells. And what we found was that we saw the formation of aggregates, and these aggregates would increase in size. We counted them for up to 28 days, and we could see, you know, a sort of much larger aggregate at the end of the culture period. And then when we looked, we did imaging of these aggregates, we could see structures within the aggregates. You can see here that look very much like follicle-like structures. And we could then dissect these structures out and then grow them within our multi-step culture system to see if they would, if they hadn't formed oocytes. So you can see here, this is our multi-step culture system. So we could take our aggregates essentially instead of the microcortex, dissect out any follicle-like structures and then grow them individually and then examine any of the oocyte-like structures that had been formed. And what we found with these, when we've combined them with the somatic cells from the adult human ovary, we find that we can obtain complexes that look like oocyte, granulosa cell complexes. And then when we stain them, we can see that they have a marker for the oocyte, DF9, so they do indeed look like oocytes. Now, of course, that doesn't mean they're functional oocytes because we need to test their meiotic potential. The next step that we're carrying out is to refine what we aggregate the OSCs with, so aggregating them with a purer population of the ovarian MSCs, so our cell lines. And essentially the experiments that we're carrying out is to define the media that will support both the OSCs and the MSCs So working out, you know, the combination of media that will lead to the improved development of these aggregates. And we're also looking at whether it's a contact, so it requires, if it's diffusible factors that can induce, you know, the OSCs to develop into oocyte-like structures, or whether contact's required. And then we're looking at the aggregation assay, where we just use the cell lines that we've produced and see how efficiently and effectively they form aggregations. And they do form aggregations, but we've got quite a long way to go to, you know, evaluate this process and each of the cell lines. In addition, we're co-culturing these ovarian mesenchymal stem cells with our ovarian microcortex, so our multi-step in vitro growth system, to determine whether we can improve the activation of primordial follicles and the health of the growing follicles. So that's another use of these OMSCs. So in summary then, the adult human ovarian cortex is a source of mesenchymal stromal cells, and functional cell lines can be produced, and the capacity and the potential of these cells to promote oocyte formation or development in vitro is currently being tested. So hopefully we'll have more data on these cell lines, and maybe at the next meeting. So finally, I'd just like to acknowledge the people who do the work. Dr. Yvonne Clarkson, postdoc in the lab, who's done a lot of this work in terms of driving the cell lines, along with Kay Samuels, who provided us with the pancreatic cell lines, and Minda Wu, Tara Doherty-Eagles, and Emily Hertzstein. And Marie McLaughlin, of course, for developing the multi-step culture system, and Joanne Grosbois, who's been instrumental in the culture work in our lab as well. And my collaborators, clinical collaborators, Richard Anderson, Hamish Wallace, Jude Tong, and a long-term collaborator who does a lot of our imaging is David Albertini. MRC, of course, fund this work, and we couldn't do any of this if it wasn't for the nurses, Norma Forson, who recruits our patients, and the patients who kindly donate their ovarian tissue, and the clinical teams that support this. And thank you all for your attention.